Hi, my name is Carly Seligman, and I work in the Raper Lab studying Zebrafish, olfactory sensory neuron axon guidance. So in order for the brain to understand olfactory sensory information from the outside world, it organizes distinct odorant experiences into a spatial topographic map on the olfactory bulb through the use of olfactory sensory neurons, or OSNs, that line the olfactory epithelium. And each OSN monolithically expresses one and only one odorant receptor. And ultimately, these OSN axons will project to odorant receptor specific neuropil regions called glomeruli in the olfactory bulb. But prior to this segregation, the axons will initially project to large, distinct, identifiable neuropil regions called protoglomeruli that are innervated by OSNs expressing closely related ORs. Now, genes for Robos and Slit proteins are highly conserved across all species, vertebrates and invertebrates. And it was soon concluded that roboreceptors bind slit ligands to regulate axonal guidance and prevent midline crossing. So while it is understood that certain members of this class, for instance, Robos 2 and 3, are important for olfactory sensory axonal guidance, the precise mechanisms of distinct interactions between members of this class are still unknown. So my project was to investigate the interactions of distinct Robos and Slits in OSN targeting in the developing olfactory system of larval zebrafish. So we utilized an F0 approach as described by Kroll et al. That entails injecting 3 synthetic guide RNAs and Cas9 complexes, together known as ribonucleoproteins, or RNPs, into single-celled larval zebrafish, and that these are offspring of 2 transgenic lines, to create an F0 knockdown zebrafish. These RNPs have a high frameshift mutation rate and are known or proven to cause 90% biallelic knockdowns. Therefore, they cause sufficient knockdown of the gene of interest, and any resulting misprojections can be visualized through the fluorescent labeling of the transgenic lines. We found that Robo2 knockdowns demonstrate striking phenotypes that include severe midline crossing as well as ventral and posterior misprojections. We also found that Slit1A and Slit1B double knockdowns demonstrate a significantly elevated rate of ectopic misprojections. And we thus concluded that Slit1A and Slit1B together suppress OSN axon escape from the central zone protoglomerulus, and that Robo2 is likely to be required for preventing midline crossing during development.